in most systems that get them out there all the time. Fish tend to lose growth, and therefore you have problems with production. You have increases in secondary mortality from infections and various other things. The problem with this is that long-term it really threatens the sustainability of industries. You're always losing this little bit of extra growth that means that the fish that you've chosen, fast growth, just isn't quite growing as quickly. It's a real problem for the farms. And at least for most farms that get problems with monogenians, they're relatively hard to control. And I guess most of my talk is going to be about why they're hard to control and how you can make it a bit easier. So, obviously, there's argument about this, but in aquaculture terms, the division between the monophystic cotyledons that feed on mucus and epithelium and the polyphystic cotyledons that feed on blood is quite useful, both because of the effect that they have and because of aspects of their biology for management. There are a number of parasites that we've been working on the last little while, and there's a few systems that we've been using. So we've got yellow-tailed kingfish, and they get a skin fluke Benedenia cereoli, and cereoli, and in Japan that's largely replaced by Tetraxine heterocerca. We've been looking at barramundi with a series of diplectins on the gills, the genus Laticola. Lepidotrimma mediana, which is on silver perch, which I won't speak about very much because my student James will be giving this whole talk about that later on. And recently we've been holding some yellowfin whiting broodstock at my research institute, and they became very heavily infected with a microcondyline. Okay. So the important thing with managing monogenians, for starters, is to understand some of these life cycle parameters. Where am I supposed to be pointing this stuff? It helps if you turn it on. Okay, so the life cycle parameters that we're really interested in are the growth, development, sexual maturity, and fecundity of the adults, their egg-laying behaviour, the fate of those eggs, and the behaviour of those eggs. They're passive particles, but they're interesting because they have this filament usually which changes the way that they operate in the water. And from there, their dispersal. How they hatch and the influences of temperature and salinity and other environmental factors are really important. And the temporal factors associated with hatching rhythms and how that influences the environment and infection is also quite important. So the first thing that we found was a real problem was that most farms didn't actually know how many worms their fish had. And with Andy Shin and colleagues, one of the things that we developed was an automated counting system called the Beast. And you can tell that this is now quite a while ago because these were cutting-edge machines when we did this work, and they kind of look a bit laughable now. Basically, the Beast works by obtaining a sample of worms from a yellow-tailed kingfish. You use a couple of barbs, strain the barbs out, you dump the worms into a plastic tray on a flatbed scanner, and you scan it. The program is a macro that runs in a SICE image analysis software called KSRun. What it calculates is a number of measurements and ratios about the targets. It calculates ratios of those measurements, and from there it segments and separates the worms out. And ultimately what you get from this program is a series of counts of Benedenia and of Zooxapter, and it gives you a size distribution, a size frequency distribution as well. So once you know how many worms you have, from there you need to think about treating maybe. And this is sort of the traditional way of managing monogenians. Oh, we've got some monogenians, we better treat them. And I always feel slightly perverse coming to a symposium where people really like these animals because I spend quite a bit of my life killing them. Anyway, I'll get over that paradox. So the traditional way of managing these things has been a bath treatment. And in a sea cage, this is quite an operation. You put the net up, you put a liner underneath it, you pump the treatment in, you let the fish swim around in it for a while, and then you let the tub hold and go. are problems with this. You have to crowd the fish. It's obviously logistically problematic and slow. You need lots of people. Most of these treatments cause either osmotic shock or oxidative stress. This is a problem for the fish. The crowding causes physical damage. Because you have to pull this big liner up underneath the cage, it's weather and tide dependent. If the water's flowing past at 5 or 6 knots, it's like trying to hold a parachute, but the liner can pull a boat, even a very big boat, backwards. It's incredibly labour intensive, and it's expensive because it's wasteful. You get one shot with the medicine, and then when you let the liner go, it just falls into the sea. So the obvious way of getting around this is to use an in-feed treatment. You don't have to handle a crowd of fish. There's less stress and mortality. There's less of an effect on productivity. You don't need that much labour or infrastructure. If you have a fish farm, you have to feed the fish anyway. It's weather independent up to a point. I've seen wind so strong that it was blowing the pellets out of the cage. That's relatively rare. You get a slower and more diffuse environmental release. This is quite important for some of the products that have a substantial environmental footprint, and it's quick. You can do all of your cages in a day, and every fish farm feeds all of its cages every day pretty much. The first line treatment for these in-feed experimental treatments has been praziquantel. It's an isoquinoline antibiotic that's specific to flatworms. It interrupts calcium homeostasis, and you get these really rapid and strange muscular effects and tegmental disruption. In the host, it's metabolized by the cytochrome P450-CYP348 oxidase system. This is really important. There's an incredibly marked first pass effect. So if you feed a fish a bolus of praziquantel, it gets taken up from the gut. One of the nice things about it is that uptake is very good, but in the first pass through the liver, you lose 50% of the product. So it's incredibly rapidly metabolized. This is actually a problem. It's so quickly metabolized that you need to use quite a lot of product. There are some ways to interrupt that metabolism, which I'll talk about briefly later on. And there's another problem because resistance has emerged in some veterinary targets. So we've tried a bunch of praziquantel treatments, both in-field and experimentally on a bunch of different fish. One of the things that we notice here is that if you know what to do, and if you deliver, a really good dose, and you've got fish that are amenable and you can get the treatment into them, the efficacy is quite good. Probably the lowest efficacy that we've had was an in-feed treatment from James's, where the efficacy against that treatment was relatively low. So one of the things that we noticed, however, every single time, pretty much poor palatability. In this one, we actually intubated the fish, so the praziquantel was delivered by tube directly to the stomach to get around that problem. Now, we sort of thought about this for a while. One of the things that I discovered is that I would rather lick a sweaty horse than eat praziquantel. If anybody comes and studies in my lab, you'll be subjected to a very perverse system where you get used as a guinea pig to taste things. But I participated in this myself, so I ate some praziquantel. It tastes terrible. It's incredibly bitter. I'm serious. I would rather lick a sweaty horse. So, why does praziquantel taste so bad? It's actually relatively simple. Commercial praziquantel is a racemic mixture of levo and dextro isomers. If you're not familiar with organic chemistry, it basically means that the molecule looks like this, and this little tail here can sit off to the left or off to the right. So we have left-handed and right-handed praziquantel. The left-handed praziquantel is both active and palatable, so this is good. If you can get this form, it's relatively palatable. The right-handed isomer is both inactive and unpalatable, but it's much, much, much cheaper to make praziquantel as a mixture. So all of the commercial praziquantel is this racemic mixture. It's not a problem for human medicine. In human medicine, you load the praziquantel in a capsule and you never taste it unless you're in my lab and I make you eat a spoonful of it. So we thought about some ways to make this better. You can mask the praziquantel with attractants. Incorporate the product into a pellet, and this is illegal in Japan, unfortunately. So one of the things about kingfish is that the industry in Japan is huge. So moving it's probably not such a good idea. The other thing is microencapsulation. This is a process where you put a microscopic coating around granules of the product. So we obtained microencapsulated praziquantel product from a pharmaceutical company, and we fed it to the kingfish. We used controls and 2 doses of praziquantel this is an incorporation rate. But what we noticed was that over time, even the microencapsulated products, the fish were stopping eating the pellets to the point where we had to stop the experiment here because the fish just stopped eating the pellets altogether. When we put them back onto a normal dose, we saw a bit of bounce back feeding, so there was a bit of overconsumption the first day. But it basically took 3 days for the fish to renormalize. So this stuff tastes bad enough that it teaches fish not to eat food. So, like I said, it tastes really bad. And microencapsulation doesn't seem to necessarily be a good way of stopping the praziquantel from tasting bad to fish. So the next thing we thought is maybe we can develop some anisomeric praziquantel. Maybe there's some way that we can resolve the pure left-handed isomer. And I thought about maybe some process of resolution where we separate the 2 isomers using chromatography or stereoablation. There's a range of chemical methods that you can use. And then the other one, probably more simple, is some kind of process of asymmetric synthesis. And the nice thing about this is that you can possibly start with relatively inexpensive starting materials. So I was sitting in my office one day, scratching my head, thinking, I really need to get onto this, when I got an alert, and these very, very kind and talented people in Germany published a resolution of praziquantel paper in PLOS One. And the nice thing about having some links to metry in human medicine is that a lot of the time your problems will be solved for you by other people. This is very elegant, this is a very simple synthetic pathway that produces pure left-handed anisomeric praziquantel. I'm in the process of having some of this manufactured right now. So while we were thinking about this and trying to get some praziquantel into the fish, we thought, well, there's all these other antimythics out there, we should try some. So the bantal is related to fenbendazole and some of these other macrocyclic lactone antimythics. We tried it against Hemisconia circa on kingfish in Japan. One of the things that we found was that in these blood feeding groups, the efficacy is excellent. And wherever I've got an efficacy value in here, there's also a statistically significant difference between controlled fish and the treatment fish. So these are really good treatment values. You know, apart from this one, we've got 100% removal of the flukes. The problem is that these fish also had Vanadenia cereoli, and when we fed that product to them, there was no effect whatsoever. There were no significant differences in intensity of the flukes between controls and the treatment groups for Vanadenia cereoli. So we have an excellent treatment for the blood-feeding flukes, but we have something that Vanadenia cereoli essentially laughs at. These worms are on the mucus on the outside of the fish going, there's something nasty in there, but I'm not eating it. So, I've got some questions left about praziquantel. We've essentially paused our work on these other treatments because none of them work on Vanadenia. In terms of praziquantel, we need to understand this uptake and distribution model much better. The metabolism of this first pass effect is very, very important. We could probably halve the doses if we can figure out some way to block some of these liver enzymes that break down the products. There are some Korean papers that show that smatazine improves the effect of praziquantel in fish. And so we're looking at some other products that may be available for use in food fish that block these sodium carboxylates. Frequency of delivery is another thing that we're looking into currently. If you feed the product to the fish twice a day instead of once a day, you may be able to kick up the dose of plasma and get some product out into the mucus. Parasite behavior is a big one. We've run a preliminary experiment that shows that one of the things that happens when you start feeding praziquantel to fish is that Vanadenia stops feeding, and that they can stop feeding and stay alive for quite a long period, up to a week. So this may be a real problem, and it may be at the heart of why this parasite is so difficult to treat and manage. very difficult to understand the difference between something that's clinically meaningful, how far back should I knock a population of parasites on fish, to make some difference to the management of the farm and to increase their productivity, as against a regulatory requirement, say for a 90 or 95% decrease in intensity. And the World Association for the Advancement of Veterinary Parasitology have been promising some efficacy standards for ectoparasites in fish for some time, but they haven't appeared. So if there's anybody out there on the stream who's involved in that, please hurry up, we need you. We've got preliminary environmental data and preliminary residue data, but we need to improve on those so that we know that the product is safe for use in the marine environment. We need to incorporate all of this into an integrated pest management system. What does that mean? So we've got a couple of products that we can feed to fish that kill worms. Where do we go from there? We need to identify individual management units. We need to get to a situation on a farm where you're actually limiting the success of the parasite and its life cycle, not just managing a situation when you get heaps of worms and when fish start dying. To do that, you need to consider the parasite life cycle and include multiple modes of treatment. It's really important to optimize husbandry. If you can fallow growing areas and separate age classes by having different sites, it will improve your management. There are difficulties, of course, in doing this. They're primarily associated with cost. You can orient the holding systems, make sure that the environment is as clean as you can, and keep your fish happy. So, if you have a situation like this is in Kyushu in Japan, trying to define an individual management unit is impossible. If you own this farm here, this guy who owns this farm here does not treat in the same block period as you, your fish will become infected from this fish. So this is a very difficult situation, and you need to develop a relationship in these kinds of places. The fish farmers tend to be individualistic, and so this is a problem. So one of the things that we've determined is that in this kind of situation, if you have Benedenia cereoli, they produce about 58 eggs per worm per hour at 24 degrees and 35 ppc. You have 40 worms per fish in a cage which has 450,000 fish on it. You have this ridiculous number of eggs. So that environment back there is more Benedenia eggs than water, basically. I exaggerate, but it's ridiculous. They're somewhere in the vicinity of 2.5 by 10 to the 4, potentially 2.5 to the power of 10 eggs per 24 hours. So you have this incredible egg density in the environment. One of the things that we've shown is that just simply by putting the cages across the current rather than in line with the current, you can decrease the distance where that egg contamination is meaningful from kilometres down to a few hundred metres. So if you get everyone in a bay to orient their cages in the right direction, you have some chance of managing them. But if you're at a farm that's downstream from another farm that has parasites, you'll get it. The whole aim of this is to interrupt the life cycle. So, start out with a situation where you have lots of adult worms, lots of larvae, and lots of eggs. This is very obvious to me, but I'm going through it because it seems to need clarification. You do a treatment in here, and if it's effective, you'll have no adult worms, but you still have residual larvae and eggs in the environment from this time here. Once you've allowed some time to pass, you get to a point where you have to have recruitment, so you have relatively few larvae and relatively few eggs in the environment because most of the eggs have hatched, most of the larvae have died, but you only have juvenile worms. And if you treat these fish again, before these worms become adults, you can get to a situation where you've interrupted the life cycle effectively and you have very few adult worms or no adult worms and very few or no larvae and eggs. So this is possible. And primarily it's understanding this kind of system and the practicalities associated with treating farms in this kind of way that makes monogenium difficult to manage. So you need to treat strategically. The block diagram represents a model that we've developed to help farmers manage their parasite burdens. These are treatment days, these grey hatching eggs and larvae, and this green box is the window for your next treatment. The difficulty with this is that if the wind comes up, or someone gets sick, or the boat breaks down, and you don't treat the fish again, by the time you get to this red line, you start to get maturing worms, and the eggs start to get laid again, and the life cycle starts over, and you have to start from scratch. We can only help so far. So the other thing is that efficacy is really important. You have your treatment here. If you can treat to 99% or better efficacy, you have a much better residual effect long term after your 2 treatments here and here with red arrows. Sorry, Andy. You have a much better long-term effect on the parasite populations. It's more than an order of magnitude. So making sure that those treatments work really well and those big numbers around 100% are really important for long-term effective management. There's also ways that you can manage fish specifically to control flukes that don't involve chemical treatments, apart from these ways of laying the cages that cross the current. This is from work that Sho Shirakashi and his colleagues did in Japan on amberjack and other Seriola species. They were individually pit tagged and they were held at 0 to 4 meters and put here a quarter-shell cage which was infected with Neobenedenia duralline, so another skin fluke. At 2 meters, you see an 80% reduction in worm recruitment, and at 4 meters you see a 95% reduction in fluke recruitment. And this is simply because the Onchomericidia are positively phototactic. So if you look at fish behaviour, you might have a cage that's got 450,000 fish in it, but if you hold them so that they can get to the surface, most of the time they only utilise the top 2 to 3 meters of the cage. Unfortunately, that's where all the parasite larvae are, because the parasites know that they've evolved with these fish. They know that they prefer to be up near the surface. So if you keep them at depth, you can decrease the reduction, you can decrease the rate through reinfection. And that's from Shirakashi 2013. There's another method here. You can use shading, and this is commonly used in red sea bream, Thai in Japan. The reason that they do it in Japan is so that they come out this nice red colour instead of this kind of blacky copper colour. One of the things that we noticed is that these fish that were shaded became much less heavily infected with skin flukes. These fish are subject to Benedenia seki in Japan. So this sort of shading system was investigated by Shirakashi and his colleagues. They held some fish in cages near another cage which was a source of infection, put them in the water, they hung the cages there. They left them for 48 hours and then they counted the number of parasites that recruited onto each fish. They did 3 trials of alternating shaded cages. So properly controlled wasn't just an arrangement effect. It wasn't just an effect from where you put the cages relative to the source. And one of the things that we found is that in these experimental cages with shades, you get a 70% better decrease in fluke load. And this is the reference for that. It's another Shirakashi paper. So if you can combine strategic treatment, arrangement of cages across the current, with depth management and shading, you may be able to get away with a very, very low level of treatment. And this is the future of management. Working around the parasite biology so that you can minimise your use of chemotherapeutics, maximising the simplicity of the system and getting really good production outcomes without using much chemical. One of the other things that we're working on, we're just starting on this, is looking at settlement cues. We're using protein fractionation. So you take a sample of fish mucus, put it through a fractionator, it sorts them all out. Follow this up by using an Onchomericidium bioassay on the fractions. What you can find from that is which fraction of the mucus actually attracts the monogeneins for attachment. And our hope long term We can identify the key chemicals in fish skin that actually are responsible for attachment by the Onchocercidia. One of the ideas that I've had is that when you do this, you may be able to just make a very simple trap. This sort of represents the smell of the stuff that parasites are attracted to. But you can make a very simple funnel trap. I've done this with fish mucus in quite big systems. And if you have a fairly tight funnel, you can attract the Onchocercidia in quite effectively, and they don't find their way back out again. So, long term, this may be another management option. People always ask me about a vaccine, and I include this slide for the simple reason that someone always sticks their hands up and says, what about a vaccine? I've talked with Kurt Bookman about this on a number of occasions, and for a really long time, what I say is, ha! I dare ya. No, seriously, this is a really, really hard target. A vaccine for a skin-dwelling monogenian is a very, very difficult thing. And at the moment, I really think it's beyond our technology. It's something to think about for the future, but for now, I say, ha ha. The other thing that I always get asked at workshops like this is, oh, what about insert name of magical compound? Apart from praziquantel, almost nothing works. When you feed it in feed, apart from praziquantel, nothing we have tried works. I'm trying everything as it comes out. I've tried some experimental compounds that are used in human medicine against malaria. I have tried all kinds of things. One of the things that I have found is that kingfish are very easy to poison. And you get in a lot of trouble at your institute when you start feeding things to fish that just make them go gulp, 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 no, I'm dead. There's no magical compound. So I guess to summarize, you need to understand the parasite host system. You need to have a system for surveillance so that you know how many worms you have. You need to understand the epidemiology of the system and how infection is occurring, where the parasites are getting onto the fish, and how much of an impact those parasites are having. You can use all of those pieces of information to improve the husbandry. When you treat, you need to do it strategically to interrupt the parasite life cycles. Just dumping some product in when you're seeing lots of worms is a total waste of time. You need to optimize the efficacy for each of the types of treatment that you use. And I haven't had time here to go into it, but we've done a lot of work on optimizing bark treatments using peroxide and a number of other products. It's very important to mix treatments. My greatest fear about praziquantel being our only successful target product at the moment is that if we keep using praziquantel, eventually it will stop working. I don't know how long it will take, but it will happen. It's like these shampoo conversions. It won't happen overnight, but it will happen. We're in the process of doing a cost-benefit analysis now, so we know how much it costs to treat a cage of fish, but we don't know how much it costs to have certain worm burden on those fish. So we're measuring the impact on growth and therefore productivity, and we're balancing it against the costs of treatment. I guess the important thing here is to look at making the industry sustainable with minimal environmental footprint. And I'm very interested in this in the last little while. I'd just like to thank my institute and all of my supporting organisations, particularly the University of Adelaide and Flinders University. I couldn't have done any of this work without all of these people. I didn't do much of that stuff at all. I've just been coordinating some of it. So it's all very complicated. The reason that I have become very interested in sustainability is that I want to know what this little guy's going to be eating when he's 80. So Yolanda's not the only person to include a gratuitous baby photo in his talk. So that was really good. But unfortunately there's a symmetry to this, and since our last conference we lost one of our colleagues. And because Ian Whittington isn't here, felicitations and bushy moustaches to everyone. Thank you. Now, does anyone have any questions for Marty? What am I thinking? Seriously.
Good answer.
I think it's feasible for some of the blood parasites, but the targets are shifting, and modulating the host's immune system so that it works is very difficult. If you can't do it in a cow with a cattle tick, and there are no really effective vaccines for parasites in cattle that live in their blood, it's very, very difficult to imagine how you can do it effectively. And I mean, with decent efficacy and with manageable cost in a fish system. So,
do you have any idea what the effects coming from the fish virus are? Is it like in snail ISM system, like some over there called like marathons? Do you like explain to things?
I can't reveal too much right now, but yes, it is a glycoprotein. They're very difficult to synthesize, and I won't be happy that I've got the right thing until I've got a synthetic validated product that I use this against. Anybody else?
Shading the lab cages is effective to reproduce the egg production of parasites.
No. It doesn't decrease egg production. It decreases recruitment of the parasites amongst the fish.
But where do you have the fish activity? Fish normally the fish inside, but when you shoot the lab cages.
Well, with red sea bream and with yellowtail kingfish, there's no decrease in growth. There was no increase in mortality.
In the rural area, for example, in the rural area, a lot of the packages or all categories they're all covered with nets. The fish is really goes out. Okay, during the day it is live, but I don't know something about it.
Yeah, we have done it long term with at least 2 species with no negative effects. And we have been monitoring for them.
Yeah, in China actually it's quite common.
Yeah.
They just cover during the day and they cover packages.
Yeah.
That's actually
Yeah.
Maybe they haven't realized it's a way to prevent parasite infection.
Yeah, well, I mean, they're probably doing something that's improving their parasite management without knowing.
Yeah.
But it does make a lot of these species much more visually appealing in the marketplace, so it's got multiple benefits.
Yeah, although this parasite is very common, but normally when we get the lab cases, we sample the fishes. It's not easy to get lots of only a few for each of the fish specimen. Each individual host species. We only get 1 or 2, but every time we went to the field, so that's why when we ask the government for some funding to do research, yeah, it's a parasite, but not always harm the fish.
Yeah.
Yeah, that's what I'm thinking about. Maybe they shave the labs,
Yeah.
the lab cages, so that they don't know, but it prevents the parasite infection.
Okay, great. So we can move on to our next speaker. Thanks. So next we have Amit Tripathi from Rajiv Gandhi University in India, talking about the invasive potential of parasitic monogenoids by the aquarium fish trade. This is a very important route for the passage of fish around the world, so it's very interesting.
Could you use a microphone?
I shall be speaking on invasive potential of parasitic monogenes, which have been translocated worldwide via the epidemic trade, and I will be making a special reference to India. This is the topic which I have been working on past maybe 2 years, and I'm still in the process of revising and improving my ideas and concept about it. The aquaculture, which was started around 2000 years back during the Chinese and Roman empires, has come a long way to become the fastest growing animal food producing sector in the world. And in the success story of the aquaculture, a very important role has been played by the introduction events of the non-native fishes, also known as exotic fishes. In fact, of the global fish production, the 17% is contributed by the exotic fish alone. However, as more and more data are being generated, many of these exotic fishes have been implicated in the loss of fish biodiversity with the competition for food and space, predation on and hybridization with the local and native fish, and then probably the most important is the transmission of pathogens and diseases. While the transmission of diseases and pathogens following the introduction of exotic food fishes have been documented very well, but those of translocated via aquarium fish trade have not received enough attention, and in particular the translocation of monogenes have not been documented very well. So I started my work first compiling a list of the freshwater monogenes that have been translocated worldwide, and I ended up with this list. This list is far from being complete because of the time and the space constraint. But what is really important here is that in almost none of these cases, the switch of the exotic monogene to the native host fish and the subsequent mortalities, if any, has ever been reported. But the case is different when we see the translocation of exotic monogenes via the food fish. There are plenty of cases well documented, and probably the most important is the Gyrocactylus stellaris, and according to King and Table, the economic losses range in excess of 500 million US dollars. And this is one of the many new threats which we have circulated in the Norway to keep the people and population aware of the Gyrocactylus stellaris. These are some of these steps of the host parasite invasion, and this I have prepared after modifying from some of our previous workers. And I must emphasize here that the success invasion of the parasite must be seen as a combined success invasion of both parasite and host with which it has been introduced with. Let us consider these steps one by one in a brief. Occurrence. Occurrence is the probability of infection, whether the infection will be there or not. And to me, this is the first and the most important stage of parasitic invasion. Now, the occurrence with parasitic monogenes is far greater from any other pathogen associated with the importation of the ornamental fish because of just one simple assertion that each species of fish is typically found infected with one species of monogenitis. This was initially given by, I think, William and Jones in 1994 and later refined and established by Williams in 1998. The taxonomic diversity of the host and the parasite is also said to be one of the important criteria of defining the invasion success. When we look at this taxonomic diversity in case of aquarium fish, there are more than 5,400 aquarium fish which are currently being traded all over the world. And as far as the number of species of monogenitia is concerned, there is no official data available. But Professor Krisky, in one of his personal conversations to me, indicated that the number of species of monogenitia described till date is already on the order of 10,000. So this huge taxonomic diversity of the monogenes and the aquarium fish traded further optimize the occurrence of parasitic monogenes in the aquarium trade. Now, the translocation to me is a function of the host specificity and also the quarantine procedures. The monogenes are said to be the most host specific of all fish parasites. And in fact, the monogenes display a higher level of co-evolution by virtue of, co-evolution, higher level of co-evolution by a co-speciation by virtue of their host specificity leads us to believe that these worms remain in very close contact with the host, and that's why they will be co-transported along with their host. In other words, these parasites will not be lost during the translocation process. Among the quarantine procedures, as we know, except few countries like Australia and maybe the New Zealand, the quarantine procedures are not well defined in many countries, and if they are defined at all, their implementation is not good. And even if the fish has been performed in the quarantine facility, the fish could still be harboring the monogenes because of the two reasons. First, the morphotaxonomic detection of the monogenes, especially those of the freshwater ones, is very difficult. And second, the infected fish, the fish infected with the parasitic monogenes, display a very little or no histopathological effects. Like excessive mucus production, pale gills. So what happens, as you can see, the killing of the fish in the Norway by salmon salads was long attributed to the industrial pollution, while actually it was caused by a monogenoid. We all know about it. Now, release. Normally, the aquarium fish are not believed to come into the contact with the farmed fish, and therefore it is assumed that their parasite will also not come into the contact with the native fish fauna. However, as again the data are being generated, you can see up to 40 species in US, North America, up to 22 in Australia, and up to 18 in India. These are the fish which have been found reproducing in the open waters. And there are several regions have been cited, like the aggressiveness of the fish and maybe the large size of the fish, because of which both retailers and the shopkeepers routinely are releasing these aquarium fish back to the open waters. But I can point out one very strange reason in India. In India, as you know, it is a very spiritual country, lots of gods are there, and the chief of the god is Lord Vishnu. And what happens? That the fish in India is supposed to be an incarnation of Lord Vishnu. You can see the lower part of this god is made up of the fish. So what happens that in India, the fish is considered to be a very sacred animal. So what happens that many people release the fish back to the open water to gain the spiritual mileages. So data is not available how many fish are constantly being released, but I believe that more or less thousands of fish are constantly being released every year. And also I would like to point out, in relation to the aquarium fish, what I think that it is not very easy to go to the river spot and catch the fish with a complex scientific catching devices. So what happens, people go directly to the pet shops, purchase the fish and release them back into the open water. And it doesn't matter for them whether the fish was exotic or the native. And this is the list of the exotic fishes which have established their population in the open waters of the country in India, 6 families. Now coming to the establishment, there are three basic concepts which are often cited in favor of establishing the host fish and their parasites. Probably the most important is the propagule pressure, which is often defined as the number of fish introduced and the number of times they are introduced. And so what happens, the fish which is traded more is more likely to have established its population in the open water. And this is working very well in India, where against the 7 exotic food fishes, 18, which I shown you on the last slide, have already established their population in the country. And also, this propagule pressure forms a kind of vicious cycle. The growth of the aquarium trade typically depends upon the import and export of fish. So when there is a growth of aquarium trade, that means there is more export and more import, and that means there is more propagule pressure, which in turn lead to the establishment success. Now, among the biological attributes, I can cite at least 2 very important biological attributes of monogenes. One, they have very short lifespan, which coupled with their hermaphroditic nature, which allows them to reproduce rapidly and directly reinfect the host. And one example I can cite here, this fish, pronunciation may go different, but I call it Tautoga onitis. This is a falciform fish, and this fish developed a massive infestation of a Gyrodactylus species, which was not a normal parasite of this species. after being kept in the New York aquarium just within a month. So within a month, it developed a massive infestation with this parasite, which is not a normal parasite of Taldoga ornitis, a perciform fish. Besides, there is one more reason I can cite, that margins have the direct life cycle. I mean, it could be very difficult for a diagenine parasite to find both the snail and the fish. So this is another reason that the establishment success of the exotic margins is optimized. About the global invasion history, the data is really not much available with me, but still there are certain hints that those fish and the parasites which have established their population in one country may also establish elsewhere. For example, regarding the monogenes, this is the distribution of the Pseudodactylgyrus anguilli and guinea. They have established their population throughout the world. So such a species should be taken care of. Regarding the fish, the 18 exotic ornamental fish species which I shown you on the last slide, they have established themselves in one or another country as well. Now, this is probably the most controversial part, that whether these exotic monogenes, once transported, will be able to make a switch to the native host or not. A group, of course, a larger group suggests it will not, because of the high host specificity of the monogenes, and also because of the phylogenetic dissimilarity of the exotic and the native fish species. But then, under the aquarium conditions, I believe, and it has been documented very well, that the host specificity of monogenes is seriously diluted. And in fact, according to Bakke et al., I think it was 2001 paper, that monogenes go on infecting as many fish as possible. And the best example is probably this capsulate Mubanidina milani, which in New York aquarium, again, has been found infesting as many as 21 families of fish, the single capsulate monogene. And then there are, yes, one more thing I would like to add here, that the very concept of aquarium, where the aquarium fish from multiple source nations cohabit, provides the monogenes an ideal opportunity to infect others, including the native ones. And should the native fish infected with the monogenes be released into the open waters, again, it has the possibilities. And even in the open water, there are a number of cases which have been showing a very low host specificity and may therefore be a candidate for transportation. This is the probably the poor part of my presentation, but this is not my fault. Actually, not much work has been done on this subject. So the actual damages open to in any area is open to question. And the part of the problem is, of course, that we do not know how to determine the mortalities in the wild conditions. But nonetheless, given the historical instances of the damages caused by the monogenes of the exotic food fishes, we should be aware of this thing. So in the conclusion, the aquarium fish trade is a perfect gateway for the translocation of the monogenes, and with the expansion of the aquarium trade, which we all agree is actually growing, there will be a more increase in the global transportation of the monogenes worldwide. However, we do not have any substantial and quantifiable data yet, but nonetheless, again, given the historical examples of the monogenes in the food fish translocation, I think there are potential risks always associated with it. So thank you very much. Some of these ideas have been published in this article, so in case you are interested, you can have it. Thank you very much.
We've got time for a couple of very quick questions. Does anyone have any questions for Ahmed?
Don't you think that hybrids may act as a bridge between the internal species and local species? But hybrids, yeah, they play a role in the
That's why it's doing self-referencing ad conference. Yes. think what he's suggesting is that if you have genetic changes in the host populations, the susceptibility to the parasites may change. And I think that happens. And I think, as Dan pointed out the other day, we're also coming to a situation where we may find that changes in climate and environment are also forcing.
So you mean that will it impact the recruits of the monogenes, exotic monogenes, this hybridization process?
Just the host switching.
Yeah. That will be impacted. So you mean there will be less possibility of the host switch then?
No, I mean that if it will species, hybridized with local species,
Okay.
this may be enriched or with parasitic or local species.
Got Louis next, and then
I mean, you showed us the photograph of the people releasing the fish to score some religious points there. Is it customary in India to do the same with turtles? To release turtles?
No, it is not. It is actually the fish, which is supposed to be incarnation of God. Hence it is a very sanctified thing.
Yeah. Because in some of the countries in the East, like Malaysia, you score a lot of points if you release turtles at the temple. And I was told, I was about to collect some turtles at the temple. They said you will lose a lot of points there.
Fortunately, this is not the condition here.
I think that sounds like suicide. Yako.
Yes, from phylogeny, we sometimes have some support for our theories. In Canada and USA recently, Virodactylus was described on frogs, bullfrog. And from unpublished data, we know that this Jenny parasite is kin of Virodactylus macrocanthus, which is original oriental weather fish parasite, Miscurnus anglicauda. So our hypothesis is that it was transferred with this host to North America, Australia, everywhere. And the parasite was in USA switching to frog, which is possible according to books.
Okay, I'm going to have to stop it there. We're starting to get animalist. So I'll stop there. Thanks very much, Amit. We have a minor change to the schedule here. We'll have one more talk before coffee. Martin's ISM is going to talk about Polystoma africanum and its internal reproductive cycle.
Good morning, everyone. This is for the host switch. In the last ISM in Cape Town, we talked about this parasite, Polystoma africanum. And there we did a redescription of the parasite. But thereafter, we made a few more observations which we are presenting in this conference. This parasite is the parasite of the urinary bladder of the common African toad. It's known to have a normal reproductive cycle in the tadpoles of the toad, and it reaches maturity in the post-metamorphic adult in the urinary bladder. Salamic adults has also reported eugenic stages of the parasite. But what was not known until now is that this parasite also has an internal cycle. course, for those who are acquainted with polystoma and its brothers, this phenomenon is well known in the Eupolystoma and Metapolystoma species. And in one member of the genus Polystoma grassae, this parasite has also been reported to have an internal cycle. In this method of reproduction, we find that eggs that are produced by pre-existing infection hatch within the bladder of the host, and they reinfect the host and establish themselves along with the adults that produced them. In this presentation, we are going to make our own observation of what we've seen in Polystoma africana. And we're going to also look at the factors that we think influence the process in these parasites, and maybe other African polystomes in which we have made some other observations. And we also observed that there's a high frequency of intestinal anastomosis in this group of parasites, and there are some remarks we also want to make about the nature of their hambrula. We collected the totes used for this experiment from two bioclimatic zones in Nigeria, in the savanna mosaic and transition forest. These are usually areas that have reduced precipitation in Nigeria. We recovered the parasites, placed them in the holding medium, 0.7% amphibian solution, and we flattened the collections, and we stained with acetylcarmine and made permanent marks, Canada pattern. In the first collection, which we actually did in 2008, we found that one of the 15 toads we collected on that location was infected with 86 polystomes. The body lengths are stated there, 2.23 to 5.23. And we believe that the ones with the larger size were the ones that produced the eggs. Of this number that we were able to measure, 55 of them, 34 were mature, 21 were sub-adult. The egg distribution in the uteri stated, and we find 45 out of 55, about 81% or 82% had intestinal anastomosis. Those we collected from the transition forest, 2 out of the 14 irregularities were infected, and one was infected with 10 polystomes and the other one with 117. This is very unusual for polystomes. Again, the body measurements for 67 parasites that we measured is stated at 3.1 to 5.1%. And 65 of this number were egg laying, only 2 in those we stained and measured were sub-adults. Again, 71.6% had anastomosis. This was our presentation, part of our presentation in Cape Town. We found very funny types of intestinal formations and anastomosis in the parasites that we isolated. Here you find, normally you don't find anastomosis across the reproductive region. It's there. There was a preponderance of the joining at this end in most of the parasites that we looked at. This is bizarre one. It has a cross, an anastomose, and a loop. I don't know what it's all about. Then, recently also, the ones we collected, they are even more interesting. The intestine does not join completely. Here there's a break. Here there's a break, but a little isthmus joining it. The same thing happens here. And these are the same. Here, only this point is joining it at the bottom. Here there's no joining at all. At the bottom here, you find there's a cross. That's what's holding everything together. This is a mirror image of this. Again, you find this is similar to what we found in the 2008 collection. The most interesting one of it is that there's a brick here, there's a brick here, all kinds of joinings that we noticed in this parasite. Anyway, we'll comment on them later. And like I said, a preponderance of undivided hamuli were found in this parasite. Only very few of them, especially the ones that were in the 5 mm range, had their hamuli behind it. But the second thing we want to talk about is the in vitro egg deposition in this parasite. We initially, for just an accidental finding, we put some polystone in our holding medium, and about an hour later, I wanted to work with them, and the petri dish was filled with eggs. And I asked my student, did you pour the eggs in here from the urinary bladder? And she said, no. And I said, then how come these eggs are here? So, I removed the polystone so that I could collect the eggs and transfer them to a new medium. In another hour, there were hundreds of eggs. Then we knew that something was happening. And so we followed it in greater detail. And we observed that these eggs were only deposited by parasites that we found in the dry season. Those ones that we collected in the rainy season did not undergo this process. And we have now observed this same process in 4 other polystomes, Polystoma eschlevani, Bayer grassei, and P. garamensis. And we'll be talking about the implication of this later. These are some of the eggs we found for Polystoma africana. We also have photographs for the other species also. We're very them for something else. We took a look at the parasites that were producing these eggs, and you could actually see this process going on real time under the microscope. The egg is released from the egg chamber, a bit amorphous, and then it goes through the process of rounding up, tanning, and then released to the exterior. And the uteri of parasites undergoing this process will contain as many as 18 eggs at a time. We'll see that in the next slide. And we did a few experiments, a few preliminary experiments, in which we incubated individual polystomes and watched what the egg production rate was. And we found it was about 21 eggs per worm per hour. And you see the uterus, and you see the process. Actually, this is going on, you could see under the microscope. You see here now, this egg is not round yet. It goes on. As they get closer to release, they get rounder and they are tanned. Okay. What we can say as now is that the intensity of infection we have observed in this study is certainly beyond what can be accounted for by the normal reproduction cycle, in which the uncooked residuum develops in the gill, matures in the bladder. The toad has to be sitting in a soup, or the tadpole has to be sitting in a soup to get that kind of infection. But in any case, it is so small that none of them can carry that kind of load. And the presence of the parasite in different states of maturity, we think, is an indication of secondary recruitment. And this pattern of distribution was also observed for Polystoma grassei by the boy and collaborator in 1977. We believe, this is our belief at this time, will maybe along the line find a way to substantiate that the in vitro egg deposition we observe is a simulation of what the parasite probably does
when the host returns to water. I'll explain this. Like I said, we think that hosts that go through a dry spell, they conserve water, and the urine becomes concentrated. And that is a trigger, no, is a sign for the policeman not to do anything. No egg production. But in the process, they are mobilizing material for egg production. But once the host returns to water at the onset of the rains, the host gets rehydrated, there's water in the urinary bladder, it dilutes the concentrated urine that was here before now, and that probably starts as a trigger that stimulates the parasite to release eggs. We believe that the eggs produced at this time are not released because the frog at this stage, because it is the onset of the rain, does not really urinate, so the eggs are not voided. And what probably happens there then is that they sit in the bladder and they hatch. In vitro, we have tried to hatch these eggs to find out whether they were viable. They are indeed viable. They embryonate at room temperature and hatch in 8 days and releases very active onconeuracidia. And we believe that when this happens in vivo, the larvae that are produced attach themselves to the bladder and develop something adult. And we think that this possibly explains the process of the internal reproductive cycle of members of the genus Polypsoma. And for those of us in the Afrotropical realms, almost all our frogs go through, except in the Niger Delta where it rains all year. All the frogs and food goes through a dry spell, and the hosts, the polystomes in these hosts that go through this period behave similarly. At this stage, we do not understand the high frequency of the intestinal anastomosis we have found, and the other intestinal conditions like the oryx. We saw in other slides, 81% in those from the Savannah mosaic and about 72% in the transition forest. Well, this is a joke. We don't know whether it has any physiological function, or they were broken because there were too many of them struggling for space within the bladder. That's why the injuries arising from overcrowding. It's also noteworthy that most of the hamuli of most of the parasites are undivided. This is actually a feature of sub-adult parasites, because in adults, the Africanus that we have described and other authors, the hamuli are well divided into a handle and gut. Since most of these parasites are small sized, they have undivided hamuli, but are egg producing. We think they are behaving reproductively like eugenics, who commence reproduction precociously. I think that's the situation. So from the foregoing, we are saying that apart from normal reproductive cycle and neoteny which have been reported, this parasite has an internal cycle of reproduction, which is triggered by the host return to water and subsequent dehydration after a dry spell. And lastly, that this phenomenon may be more widespread among African polystomes than previously. Thank you, Professor.
Thank you, Martin. Are there any questions?
Have you ever observed or even measured if hatching of onconeuracidia depends on the osmotic activity of the fluid inside the bladder?
Yeah, that's what we are trying to do. We are trying to, in the next set of experiments, we want to take the eggs and put them in different concentrations of saline. maybe starting with water, but the problem with water probably is that they will get the surface water lost. But we take a gradation of salt, and no, in water it's okay. Yeah. No, for the parasite that will not be safe because it's the parasite itself, but the eggs is okay. But we'll try that out and see what happens.
What we've noticed is that exposure to UV stimulates plants. And what we would typically do, if eggs are ready to hatch, we would take a petri dish out and expose, lift it and the lid, exposed to sunlight for 30 seconds, take it in, and then sometimes if you put it under the microscope, it's like popcorn, you know, exploding. And sometimes they won't, you know.
So could the pH is hyperosmotic and the hyperosmotic conditions?
I've tried some experiments with that, and they couldn't get it to match.
Yeah, because the question you raised is very valid. Someone has raised the issue. We want to dilute and see what level of salt will be tolerated by the eggs, and that's whether it's equivalent to what the urine might be if we dilute.
Okay, great. Well, it's coffee time. Walter, do you have any announcements?
Now we have the photographs from the group photograph, one per person, so if you want to come here, I'll be
Come and get your photo.
there when you go.
See how beautiful you all look.
And our
Sir Shalom.
local president.